Hello students, I am here with my channel Solving Questions with Garima. Here we solve questions from IIT JAM examination. So let's have question for today's video. The question is 1 mark MCQ type of question from paper of Biotechnology 2019. The glycosidic linkages in cellulose and amylose are alpha 1,4 and beta 1,4, beta 1,4 and alpha 1,4, beta 1,4 and alpha 1,6, or alpha 1,4 and alpha 1,2. We have to solve this question today. Yes, for understanding this question, we'll go through what is this alpha bonding, beta bonding, what is alpha 1,4 linkage, beta 1,4 linkage. So let's go through this first. So here on the board I have drawn the structure of glucose. This is a straight chain structure of glucose. All of us knows this structure. It is having a 6 carbon sugar and it is D-glucose. When we say capital D-glucose, the hydroxyl group on the penultimate carbon, that is the second last carbon, the hydroxyl group on the second last carbon is towards the right hand side, then the structure is known as D-glucose. If we hold the structure in this way, in hand, in this way, and if we find the spatial structure, that is the hydroxyl group on the penultimate carbon comes on the right hand side, then we call it as a D-glucose. So this is a straight chain structure of D-glucose. Now, when this straight chain structure, or when glucose, when any sugar enters into the aqueous environment, then it tends to form a cyclic structure. This is the main thing to remember. In the aqueous form, every sugar will form a cyclic structure. So this cyclic structure, formation of cyclic structure is the base for the formation of alpha or beta anomers. See here, this hydroxyl group on the 5th carbon atom. This hydroxyl group, which I have shown in a blue marker, this hydroxyl group reacts with the first carbon atom and it forms this type of structure. This we call as a hemiacetal. In case of aldehydic sugar, we call it as a hemiacetal. In case of ketone, the sugar having ketone group, we call it as a hemiketal. So this is a hemiacetal structure, and when this structure further resolves, it can form two types of structures. If this hydroxyl group, see, all our focus is on this hydroxyl group. Now the hydroxyl group on the first carbon atom. So if this hydroxyl group on the first carbon atom is at the lower side, then the form is known as alpha form. Okay? If this hydroxyl group is at the upper side of the carbon atom, in this way, so we call this form as a beta form. This is to be remembered. When the hydroxyl group at the lower side, it's a alpha form. When hydroxyl group at the upper side, it is a beta form. Now, what is the name? Alpha D glucopyranols. Okay, so alpha is okay. Just now I have understood. We have understood. Then D, then that is the D taken from here. Gluco is glucose, structure of glucose, and it is this ring, the 6 carbon ring. This type of ring is known as pyranols ring. Hence the glucopyranols. Okay. So in this way, this alpha and beta forms. These are the two stereoisomers formed due to the formation of cyclic structure of the glucose from its linear or straight chain structure. Now to understand our question, I have drawn here two structures, structure of cellulose and structure of amylose. So just now we have seen that when the hydroxyl group is present at the above side, it is a beta ionomer of the glucose, and when it is below, it is called as, it's a alpha ionomer. So now beta 1,4 linkage, it can be clearly seen now. The hydroxyl group is at the above position and there is a bonding between two glucose units in case of cellulose. Also, of course, in case of amylose also, it is a polymer of glucose only, and here the bonding is between alpha 1,4. So the answer becomes, in case of cellulose there is a beta 1,4 linkage and in case of amylose there is a alpha 1,4 linkage. Cellulose and amylose both are the polysaccharides of glucose. Cellulose is present in a plant cell wall and amylose is a part of the starch. If you remember, starch is made up of amylose and amylopectin. So out of that, the amylose is this one and amylopectin is another structure. They combinedly forms the structure of starch. So the answer for this question is option B. That is, the glycosidic linkages in cellulose and amylose are beta 1,4 and alpha 1,4. This is the correct answer. So here is the question given for my next video. Here we have to match the entries from group 1 and with that of group 2. The question is from paper of biotechnology 2019. It's a 1 mark question. So in group 1 it's given nylon, natural rubber, starch, myoglobin is given, and in group 2 isoprene, hexose, amino acid and adipic acid is given. So we have to match these two groups. We'll solve this question in my next video. Till then, don't forget to like, share and subscribe my channel and also hit the bell icon. And bye and take care.